Becky. I would now like to welcome our next speaker, Dr. Austin Gould from the University of British Columbia, who will be giving a talk titled Endogenous Retroviruses Induce Protective Host Immunity Against HSV-2 Vaginal Infections in Mice. So you can get prepared, and we can now see your screen.
Perfect. Yeah, thank you so much for that introduction. My name is Austin Guild, and my pronouns are he, him, his. I am actually a first-year MSc candidate, not a doctor yet, but I do research of the University of British Columbia, which sits on the ancestral territory of the Musqueam people, and I research of the Tokiyama lab. And today I'll be talking to you about how endogenous retroviruses provide protection against vaginal HSV-2 disease. I just want to acknowledge the two first authors on this paper, Radisha and Tian Yang. I also want to give a big thank you to the Beyond Science Initiative for inviting me to speak today. Before I get into the paper, I just want to do a brief overview of HSV-2, or genital herpes, or herpes simplex virus 2. So we know that HSV-2 is a very prevalent sexually transmitted disease, estimated to infect about 491.5 million people worldwide, estimated about 13% of the entire population, so quite substantial. We also know that HSV-2 establishes a latent infection, meaning that individuals with HSV-2 will experience reoccurring uncomfortable symptoms, such as lesions, throughout their entire life. In addition to this, patients with HSV-2 are also at a much higher risk of being co-infected with HIV, contributing to about 40 to 60% of new HIV cases each year. Our lab specifically studies endogenous retroviruses, and these are retrovirus sequences that are in each and every one of our own genomes, making up about 8% in total, which is quite substantial compared to the 2% that make up protein coding sequences. These ERV sequences can then produce polyadenylated RNA, which they can further produce whole infectious ERV particles, which have only been seen in animals, not humans so far. However, in humans, we're able to produce ERV proteins and cytosolic nucleic acids. These have then been implicated in both detrimental effects, such as autoimmune diseases, neurodegenerative diseases, and even cancer. However, it also can lead to beneficial effects, such as pathogen clearance. And this idea has not been extremely well explored in the past and actually led to our research questions, which are, do ERVs mediate protection against exogenous viral infection? In this case, we're using HSV-2 as a model pathogen to test our hypothesis. Before I get into the results, I just want to do a brief overview of the experimental design. So for the first part of the study, we're looking at two genotypes of mice, wild type B6 and TLR7 knockout mice. And these mice lack the toll-like receptor 7, which is an innate sensor that senses RNA, and had previously been described to increase the production of infectious ERVs. Therefore, we first wanted to quantify this. And to do this, we took leukocytes from both genotypes, co-cultured them with DFJ8 cells, and then stained for positive ERV envelope protein and negative for CD45 in order to eliminate the leukocyte population. And what we can see on the left-hand side in the red boxes is the TLR7 knockout mice had substantial production of infectious ERVs, where the wild type essentially had none. From here, both genotypes of mice were then injected with depot medroxyprogesterone acetate, which is a drug that puts the mice in diestrous cycle and has been known to induce susceptibility to HSV-2 infection. Several days later, we intravaginally treated the mice with HSV-2 and then monitored their disease for 2 weeks, specifically looking at their survival and clinical scores. What we found was that those mice that are expressing high amounts of ERVs, the TLR7 knockout mice, as seen in blue, had significantly lower disease scores compared to that of the wild type mice that did not have any significant ERV expression whatsoever. Similarly, we also see that the TLR7 knockout mice had significant higher survival rates compared to that of the wild type mice. So this tells us the TLR7 knockout mice do in fact have high amounts of infectious ERVs, and that this is protecting them from HSV-2 infection. From here, we wanted to see if we could replicate this immune response in wild-type mice by pre-treating them with the infectious herbs isolated from the TLR7 knockout mice and then intravaginally infecting them with HSV2. What we saw was that when we treated the wild-type mice pre-herbs with before the HSV2 infection, we do see a delay in HSV2 pathology as seen in red. And this is compared to the wild-type mice that were not treated with herbs prior to HSV2 infection. Next, we wanted to redo this experiment in a highly susceptible group of mice. For this case, we use mice that lacked an interferon receptor. And really interestingly, we still saw a delay in HSV2 pathology and lower disease scores compared to mice that were not treated with herbs. So this tells us that the infectious herbs from the TLR7 knockout mice are able to create this immune protection in the wild-type mice and a highly susceptible group of mice that lack the interferon receptor. What's even more interesting is that herbs are typically associated with a strong type 1 interferon response. However, based on the data that I present here, it appears that actually might not be necessary for this herb-mediated protection against HSV2. We then want to determine what exactly is necessary for this production of infectious herbs and the subsequent protection against HSV2. We had previously known that this production of infectious herbs was most likely due to a recombination event with the EMV2 locus. Therefore, we created a genotype that completely lacked the EMV2 locus, as well as a fourth genotype of mice that completely lacked TLR7 and the EMV2 locus. And what we can see on the left-hand side is still only the TLR7 knockout mice have significant levels of the infectious herb production, whereas the other three genotypes more or less have the same relatively low amount of production of infectious herbs. So now we want to see how the disease scores would change when all four genotypes of mice are infected with HSV2. And what we can see is still only that of the TLR7 knockout mice that have the high amount of infectious herb productions have the low disease scores, whereas the other three genotypes relatively have the same disease score and it's quite high. What this tells us is that the EMV2 locus is in fact necessary for the production of the infectious herbs, as well as for the protection against HSV2. Finally, we want to do a transcriptome analysis of the vaginal tissue from all four genotypes of mice to see if there's any specific genes that were protecting against the HSV2 infection through herbs. Through the transcriptome analysis, we determined that there was 25 specifically upregulated genes only in the TLR7 knockout mice. In other words, only the mice that are producing the infectious herbs. Of these 25 genes, we determined that the only pathways that were significantly enriched were those involved in the extracellular matrix organization. And I'll touch on that a little bit more in my future directions section. So I just want to do a quick summary and conclusion about what I've talked to you about so far today. So first, we found that the TLR7 knockout mice do in fact express high levels of infectious herbs, and that these mice were specifically resistant to HSV2 infection compared to the wild-type mice. Next, we were able to determine that the infectious herbs are sufficient to delay HSV2 pathology when pre-treated in wild-type mice, as well as the very susceptible group of mice that lack the interferon receptor. In addition to this, it also points to the fact that a type 1 interferon response is not necessary for this herb-mediated immunity against HSV2. Third, we were able to determine that the EMV2 locus was in fact necessary for the high amount of herb production in the TLR7 knockout mice, as well as the subsequent protection against HSV2 infection. And finally, through our transcriptome analysis data, we determined that the extracellular matrix organization pathways were specifically enriched in the HSV2 protected TLR7 knockout mice. In conclusion, the data that I presented to you today does support our hypothesis that the infectious herbs are mediating HSV2 disease resistance. So I just want to go over a couple future directions and what our lab is currently working on. So our main objective at the moment is trying to determine what exactly is the mechanism that is leading to this ERV-mediated immunity, specifically through how they're directly modulating the vaginal tissue. I didn't have time to show you today, but throughout the study, we were also able to determine that the ERVs were most likely not acting upon the HSV2 virions directly. Therefore, it most likely is acting upon the host. Therefore, one of the leading ideas is that the ERVs are actually enhancing the vaginal epithelium, in other words, strengthening the integrity of the tissue. This is probable because through our transcriptome analysis, we did determine that the extracellular matrix organization pathways are specifically enriched. Another idea that we're working on right now is that it's possible that the ERVs are actually competitively inhibiting the HSV2 entry receptors, Nectin-1 and HVM. And then this is leading to a substantial drop in disease scores when HSV2 infection occurs. That's actually my current project as a master's student, and I would love to talk about that more in the Q&A if you have any questions about that specifically. Down the line, we also want to investigate what exactly is the minimal component of ERVs required for this observed protection. In other words, do you actually need the whole virion that we see in the mice, or is only a piece of the virion necessary? For an example, the envelope protein. If we can determine exactly what the minimum component is required for this ERV-mediated protection, it could have a lot more applications to human health. As I had said previously, it has not been seen in humans yet that we can create the whole infectious ERV virion. However, we are able to create the envelope protein from the ERV sequences. Therefore, if we can determine this, it might have more applications down the line. And finally, down the line, we also want to investigate why do we see subtle differences in level of immune response between the TLR7 knockout mice that are producing constitutive ERVs compared to the wild type mice that we treat intravaginally with ERVs prior to HSV2 infection. One idea is that you might just need a constitutive expression of ERVs. Therefore, that's why you would only see such a robust immune response in the TLR7 knockout mice, not the wild type mice that are treated intravaginally with the infectious ERVs. Another leading idea is that you might need the ERV production in the peripheral tissues around the vagina. Therefore, you would not see as robust of an immune response when treated intravaginally with the infectious ERVs in the wild type mice. At this point, we do not have the questions to all these concepts. However, in the near future, we hope to investigate them further. Again, I just want to give a big thank you to the Beyond Science Initiative for having me speak today. Again, big thank you to the co-first authors on this paper, Radisha and Tina Young. These are the members of the Tokiyama Lab, my funding source, and if you have any further inquiries, please feel free to email me at this email address on the screen. Thank you.
Thank you so much for that comprehensive and amazing talk. We do have some questions, so we will start with the question from Dr. Fish.
It was a great talk, Austin. Thank you very much. So I just wondered whether you'd given some thought to what those key elements might be in this extracellular matrix organization that are contributing here.
Yeah, it's really tough to say. We don't know quite yet. We think that it could just be strengthening the integrity of the extracellular matrix in general and just strengthening the vaginal epithelium. Therefore, perhaps allowing less bacteria or less viruses to actually enter the vaginal tract and lead to infection. But at this point, we don't quite know what specifically that might be so far.
Okay, thank you.
I would now like to welcome Pavan Raj to also ask a question.
Hey, Austin, great talk. I just had a question about whether the prospect of investigating HSV1 along the same lines can be done. The narrative of genital herpes being caused predominantly by HSV2 is now changing just because of different sexual practices. So is that something that you're thinking about?
Yeah, no, that's a really great question. Yeah, we definitely do want to investigate HSV1 in the future. And like you said, sort of the lines between HSV1 and HSV2 is sort of becoming more and more blurred. And I think that it really will come down to sort of what we determine through what the mechanism is. As I said in the future directions, if we determine that it's through competitive inhibition of the receptors, it might be a little bit more specific because we know that HSV2 specifically uses nectin-1 and HVM in the vaginal tissue. However, if we determine that it most likely is due to a integrity, like the vaginal tissue being strengthened and the integrity increasing, this absolutely could have broader applications to all mucosal surfaces, including, like you said, the oral cavity and HSV1.
Thank you. And we do have one more question from the chat, which we will just ask out loud, so then you don't have to type it out after. But someone asked, Jacob asked, how can these findings apply to human health protection from HSV2? And are there potential combination therapies that may add to these findings?
For sure. Yeah. I mean, of course, we're far a ways away from actually applying this data to humans. But like I said before, humans are able to produce the ERV proteins. Therefore, again, really far down the line, you never know, we might be able to create some sort of therapy if we determine that it is because of the ERV envelope protein, or it might just be a intravaginal treatment with the ERV envelope proteins, and that this decreases your risk for HSV2 infection. I think at that point, that's sort of where we're at for right now.
Thank you. Well, I believe that's all the questions that we have for now. If more questions do pop up, and if anyone thinks of anything, please write them in the Q&A section. And Austin, if you just keep an eye on that section, you can type out your answer. That'd be really great.